Vosotros pedís permiso para la gente.
Well, thank you. Thank you everyone for coming. Very happy to present today's speaker as well. Myra Hughey is also a sabbatical researcher here in EBD for still a month, short of a month. We were also lab mates in Boston University a while ago, and Myra, she studied as an undergrad in Loyola University in New Orleans. Then she came for a PhD at Boston University, and then she did a postdoc at Virginia Tech with Lisa Belden. She started studying predation of arboreal eggs in tropical tree frogs, and then you moved on more, became more of a microbial ecologist, and she's now a professor at Vassar College. So, the floor is yours.
All right, thanks, Yvonne. And thank you, everyone, for being here today and listening to the research that I have to share. And I also just want to thank EBD for hosting me during this sabbatical, and Yvonne and all the folks in his lab for being so welcoming to us and really making this a wonderful experience. And before I begin, I also want to acknowledge that the work that I'm going to share with you today represents the efforts of many, many people that I've had the pleasure of working with over the years. And in particular, I want to point out my postdoc advisor, Lisa Belden at Virginia Tech, and her graduate student, Corin Jones, who's now a postdoc at UT Austin. And then also my collaborator, Erica Crespi at Washington State University. So they have contributed substantially, and members of their labs have contributed substantially to this work. Okay, in the past decade, there has been an explosion of research documenting the diversity of microbial life that inhabits every organism. And this is in part because of our strong interest in understanding the impact that these microbes can have on host health, right? So these microbes do all kinds of things, from enhancing our nutrition to protecting us from pathogens to even affecting our behavior, right? And so this framework here is one that I particularly like for thinking about these systems. In this, one can imagine a host as sort of like a little ecosystem that is supporting an assemblage of microbes that are capable of performing all kinds of different functions, right? Or we can also think of these as ecosystem services. And an important thing to understand here is that these microbes are really operating kind of with little concern for the hosts that they're living upon, and also that host ability to control the kinds of services that these microbes are providing, or what their impact will be, can also be very limited, right? So these aren't these like very tight mutualisms here, right? There's a lot of room for variation. Now, when it comes to hosting microbes, amphibians are no exception. So if you take a swab and you pass it over the skin of an amphibian and then use that swab to inoculate a plate of agar, in a few days, you'll end up with something like this. And so here you can see a whole bunch of different little bacterial colonies. These come in all different colors and shapes and sizes. And this is like one of my favorite aspects of this work, is seeing these things. And each one of these represents a single bacterial cell that came from the frog and was deposited by the swab onto the agar plate. And these are great to have for all kinds of reasons. So in fact, we've amassed an extensive culture collection of these bacteria that we've isolated from many, many different frog species, and we can use this to assay their function or for experimental manipulations. But one thing to keep in mind is that these cultures only represent a really tiny fraction of the overall diversity of these communities. And we know this because we can take another swab and sequence the DNA from essentially all the bacterial cells that that swab has collected, and use a metabarcoding approach to really more fully characterize the diversity of these communities. And so for those of you who are not super familiar with this approach, what we do is we amplify a region of the 16S rRNA gene. And you can think of this gene as a sort of barcode that can be used to identify bacterial cells, more or less to species. And so when you sequence this gene from your sample, you end up with thousands of copies of it, right? So amplified from every single bacterial cell in your sample. And then you can pool all of these copies based on sequence similarity. And then depending on which bioinformatics pipeline you use, you end up with what we refer to as operational taxonomic units, or OTUs, or alternative sequence, amplicon sequence variants, or ASVs. So you'll hear me use these two terms in this talk. And we consider these roughly equivalent to bacterial species. You can also use counts of the sequences to estimate the relative abundance of these different sequence variants in your community. And then finally, the last thing you can do is match a representative sequence to a publicly available database to get an understanding of the taxonomy, or to assign taxonomy. And so what I'm showing you here is a phylogeny representing all of the OTUs that were identified from 4 species of temperate amphibians, these 4 species of temperate amphibians here. And what I want you to take away from this is that amphibians harbor incredibly diverse microbial communities, right? There's all kinds of bacteria. And that's just talking about the bacteria on these frogs. Okay, and so now that we have these tools, we can start asking all kinds of interesting questions about what kind of variation do we see in these communities, and what are some of the factors influencing this variation, and if there is variation, does it matter? And so today I'm going to share some of the work that I have done to address these questions. So first, let's just give you an overall impression of what kind of diversity we're talking about here. And for this, we'll start with a single amphibian, a typical frog. In this case, we're going to look at a spring peeper. And what I can say is that a typical spring peeper microbiome harbors hundreds of OTUs. So a single frog is going to have hundreds and hundreds of these different kinds of bacteria on its skin. And here I've summarized these OTUs at the genus level. And so I'm showing you the relative abundance of sort of the top 10 genera. But the other thing that I want you to notice about these communities is that they're often highly dominated by one or just a few OTUs. So this orange bar here with the hatch marks in it represents a single OTU in the genus Sanguibacter. And it's not really important exactly what this is, but what I want you to take away is that this one single ASV OTU represents about 20% of the diversity of the community. So now we can expand out and look at 13 other individuals from the same pond. And here what I want you to take away is that many of these individuals' bacterial communities are relatively similar, right? So we see that same dominant OTU in most individuals and fairly similar relative abundances. But we do have some oddballs that show up. And then we can expand out even further with the results of a survey that I conducted across the state of Virginia when I was a postdoc. And here I sampled 9 sites, some of which were in the Appalachian Mountains and some of which extended out onto the coastal plain. And when we do this, we begin to see even more variation. So here are these 9 sites, presented in order from west to east. And one thing that I want you to notice is that we can even get a complete switch in the dominant OTU in these systems. So here we go from a much higher relative abundances of this Sanguibacter that I just talked about to a different pink OTU, which happens to be in the genus Bordetella. Okay, so here's the kind of variation that we can see within a species. What about when we look between species? First, I'm going to compare spring peepers to 2 other sympatric species, American toads and bullfrogs. But before I show you the results, I want to introduce another way that we can show these data. And that's through a technique called non-metric multi-dimensional scaling. And this is an ordination technique that attempts to represent the pairwise dissimilarity between samples in a very low dimensional space, usually 2 dimensions. And so in these plots, each point here is going to represent the bacterial community of a single frog, and then points that are closer together indicate individuals that have bacterial assemblages that are more similar than those that are farther apart, right? Okay, so now we'll take a look at the data here. And what we can see, we have spring peepers in pink, we have American toads in gold, and then we have bullfrogs in green here. And what I want you to take away from this is that individuals of a given species tend to kind of cluster together. They have relatively similar microbiomes. Sometimes individuals of different species overlap quite a bit, but at other times they're quite different, right? Now, just for fun, we can add some tropical species on here too. So here we have red-eyed tree frogs, pantless tree frogs, and then this is a robber frog here. And now what I want you to, now what we can kind of take away from this is that we have big species differences, but maybe we also have some environmental influences here, right? So the tropical species are clustering separate from all of the temperate species, and species with similar niches tend to overlap more. So we have the two arboreal species overlapping more and kind of separate from this species, which is more of a leaf litter frog. And then we've got two more terrestrial species clustering distinctly from a more aquatic species here. Okay, so big takeaways from what I've presented so far. Amphibian species tend to harbor recognizable bacterial assemblages, but we can have fairly extensive within-species variability. And what this kind of indicates to me is that probably both intrinsic and extrinsic factors are influencing the variation in these communities. Which leads us nicely to our next question here, which is, what are some of these factors influencing this variation? And so, before I jump into this research, I just want to give you a framework for thinking about how these communities assemble, starting with when a little frog egg is first laid. We can think of assembly as occurring in two stages, arrival and establishment. During arrival, microorganisms are going to be making their way to the egg, making their way into the local community, right? And for host symbiont systems, this can happen in a couple of different ways. We can have transmission of microbes between hosts. So for example, you could have vertical transmission of microbes from parents to offspring. And then we can also have dispersal of free-living environmental microbiota to the host, right? Now, once those microbes have arrived, they may establish or fail to persist on the host. And establishment is going to depend on a number of things. First, it's going to depend on the conditions of the host. So sort of, is the host environment suitable for the microbe, for microbial growth? And then also on interactions between the microbes themselves. So you can have very strong competitive or even facilitatory interactions within these, facilitative interactions within these communities. Now, throughout the life of the host, these processes are going to continue to operate, right? Although their relative importance may change. So, for example, we could imagine that host microbe interactions are going to be likely to change as the host immune system matures. Now, most of the work that we have done so far has really just focused on sort of one aspect of this, which is dispersal. And specifically, we've conducted experimental manipulations in order to understand how different environments can influence the assembly of bacterial communities early in the life of a frog. And so I'm just going to give you a couple examples of the kinds of experiments that we have done. In this first experiment, we conducted a relatively simple manipulation of the rearing environment of spring peeper eggs and larvae. So we collected adult spring peepers and allowed them to breed. Some of them we placed into sterile environments in the lab. And then others we placed into outdoor enclosures. And so for these, we bought these big plastic bins, cut off the bottom, stuck them down in the mud, threw a pair, set a pair gently in there, and then put the lid on and left them overnight to breed, right? And so for the ones that were laid in the lab, we took samples of eggs the morning after they were laid. And then for other eggs, we placed them into individual containers to develop. And then we sampled the tadpole communities 2 to 3 days after hatching, so once the yolk was resorbed. For those that were laid in ponds, we also took samples of eggs the morning after they were laid. And then for others, we put them into mesh enclosures out in the pond. And for these, we also took samples of the tadpoles, but we waited until 1 month after we had put them in those enclosures, so a much longer time period. And so what did we see? Maybe not surprisingly, we found that eggs that were laid in ponds and also larvae from ponds had much higher, much more diverse microbial communities than those from the lab. And so this suggests that if amphibians are given the opportunity, they'll also recruit these microbial, or these free-living environmental microbes into their symbiont communities. We found that individuals from lab and pond, here represented in black and gray, had distinct compositions, and that the microbial assemblages from eggs and tadpoles were also distinct, suggesting that there's some developmental effects there, or developmental changes in the microbiome. And what was more interesting to me, though, was that we saw that individuals from the same families tended to cluster together. So here I'm showing you the exact same ordination. I've just recolored the points to indicate which pair of frogs the individuals came from. And what you can see is that all the points of a given color tend to cluster closely together. So this was really interesting because it may support a role for vertical transmission of microbes. And then what I think is most interesting are actually these samples here, which were those samples that we, or were those tadpoles that we sampled 1 month after we put them into the ponds. So we still see these, that individuals from the same family tend to cluster together. Which is interesting because it suggests that these early life experiences can have long-lasting impacts on the dynamics of the assembly dynamics in these communities. All right, so along with, this got us wondering exactly how much can we manipulate these communities based on what we expose them to, or in what order we expose them to different microbes. And so we set out to do some even more manipulative experiments, exposing eggs and larvae to different kind of synthetic communities of bacterial isolates that we had. And we've done this with 2 species now, but I'm just going to present the results from spring peepers for the sake of time. And so how do we do this? We again collected pairs of spring peepers and brought them into the lab to breed. We separated the eggs into individual containers, and then We inoculated the water that they were in with one of 3 different kinds of bacteria, which are represented by these different colors here. And the idea was that by inoculating them with this one single isolate first, we were sort of giving it a head start to colonize the embryo. We left them in this inoculum for 24 hours before removing the liquid and then replacing it with this 5-isolate community, which included all 3 of those isolates that we had used the day before. We also included a couple of controls, one that was where eggs were exposed to water on both days, and then one that received water for the first inoculation, and then the full community for the second. We then sampled the bacterial communities after a couple of days once yolk resorption was complete. And so what did we find? So here I'm showing you the average relative abundance of these 5 different isolates from all of our treatments. So we have our 2 controls on the left here, the water treatment and then the water plus community. And then on the right here we have our treatments where we gave an isolate a head start. So either this P1, which is in green, the Xanthinobacterium, which is in purple, or this P2, which is in yellow. And so the first thing that I want you to take away from this is that, yes, we can get these bacteria to grow on these tadpoles. And so that was really exciting. We can establish multiple species of bacteria into these early tadpole communities. And then the second thing I want to highlight is this purple Xanthinobacterium here, which is the only isolate that increased in relative abundance in response to being given priority. And so what this demonstrates is that for at least some of these isolates, it does seem that we can use arrival order to aid their establishment. Okay, so some things to take away from this part of the talk. Parents and the environment are both likely sources of microbial symbionts. Vertical transfer, among other things, there's lots of factors that we can talk about here that are going to influence the variation in these communities, may account for some within species similarity. And then variation in source pools and arrival order may account for some of the variability that we're seeing here. Okay, so I've told you a lot about the extensive variation that we can see in these communities and about some of the processes and factors that may be contributing to this variation. Now I want to talk about whether this variation matters, right? Because that's really, from the host perspective, that's what's really important. And we think about this question a lot in the context of disease. And that's in part because amphibians globally are threatened by a number of pathogens, including 2 types of chytrid fungi, which infect the skin. One is a more frog-oriented one, and then there's a new one that's been recently identified that seems to infect salamanders more. And then the other big pathogen that's emerging are rhinoviruses, which I'll talk about a little bit more a little later on. And so just to give you some perspective on what these pathogens are doing, this is a recent review that highlights just how impactful the disease chytridiomycosis has been on amphibians. And so here's a quote from the study. We conservatively report that chytridiomycosis has contributed to the decline of at least 501 amphibian species. This represents the greatest documented loss of biodiversity attributable to a pathogen and places BD among the most destructive invasive species, comparable to rodents and cats. So that's pretty significant. And this is just talking about one of those pathogens, Batrachochytrium dendrobatidis. And so where do microbial symbionts come in? Well, there's growing evidence that symbiotic microbes may play a role in protecting their hosts from these pathogens. We know that many of these bacteria are capable of producing antimicrobial metabolites that can inhibit pathogen growth. And other labs have done some experimental work showing that if you reduce or augment the microbiota, you can change disease outcomes. So that really provides some pretty strong evidence that they're doing something. However, we still don't fully understand what makes one microbial assemblage better than another in terms of pathogen defense. And so to look into this, we have conducted experimental pathogen exposures with a number of different species now, and including 1 temperate species and 3 tropical species here. And so in a nutshell, what we do is we collect frogs from the wild, we swab their skin to characterize their bacterial communities, and then we expose half of them to a pathogen, in this case chytrid fungus, by putting them in a little bath of zoospores, like this little guy right here, and then monitor and then swab them for several times thereafter to record what happens. And so I'm going to show you the results for just one species, Crogastrophyt singeri. But I will say that the outcomes for different species were very different. And so if this is something you're interested in, definitely ask me about it later. First, I'm just going to show the infection results. So this is infection intensity over the course of the experiment in control and exposed animals. And what this just confirms is that exposed animals became highly infected. And in fact, for this species, which is typically seems to coexist fairly well with chytrid in the wild, the levels of infections that we saw were surprisingly high. And we even had 4 deaths, so 4 individuals that started showing symptoms of disease and that died over the course of the experiment. And so one thing we asked was if there was anything different about these individuals' microbial communities, bacterial communities. And when we compared individuals that developed high infections with those that did not, we saw kind of subtle differences in the relative abundance of a few different ASVs that were present in their communities. What I want to really draw your attention to the second one, Pseudomonas, which tended to occur in higher relative abundance on individuals that did not develop severe infections. And this is something that we found pretty interesting because we also saw this pattern on a larger scale when we compared the skin bacterial assemblages of the species from areas where we knew BD had been endemic for a long period of time with this one site that we were fortunate enough to sample before it experienced the arrival of BD. So we have 3 BD endemic sites and then 1 BD naive site. And what we again saw was that in these sites that had where BD was present, we saw much higher relative abundances of the genus Pseudomonas compared with that one site that was BD naive. And so to me, what this suggests is that perhaps the presence of BD is sort of selecting for this genus of bacteria in these symbiont assemblages. Okay, I have one last story that I'm going to very quickly try to share before I wrap up. So for this, I'm going to turn to yet another species. These are wood frogs. And these frogs are common throughout the northeastern US and Canada. This species is susceptible to the viral pathogen ranavirus. And in the northeastern US, we sometimes see these massive die-offs of larvae as a result of infection with this virus. So in wood frogs, disease severity and die-offs are strongly associated with tadpoles that are nearing metamorphosis, so much later developmental stages. In tadpoles in earlier developmental stages, ranavirus activates the HPI axis and induces both stress and immune pathways that help the animal fight infection. However, in larvae that are approaching metamorphosis, activation of the stress response causes these larvae to shift energy away from immunity and towards hastening development. So instead of investing in immune function, they just try to get out of the pond, right? And that works for animals that are trying to escape other kinds of stressors, so something like pond drying, but for ranavirus, it just makes them more vulnerable to infection, and so they often succumb. Now, we also know that tadpoles that are exposed to elevated salinity are even more likely to die from ranavirus. And this is because salinity appears to suppress the immune response even further. And in cold regions of North America where wood frogs live, salinity is a real problem, right? So we have lots of use of de-icing salts to keep roads safe, but then that runs off into aquatic systems and increases freshwater salinity. And we know wood frog ponds are no exception. So this is a figure showing the conductivity of ponds measured at varying distance from the side of roads. And what we see is that the highest salinities occur in ponds that are much closer to roads. But that's not the end of the story. So interestingly, salinization has also been associated with increased productivity in these systems. So it causes the proliferation of algae. And algae is a really high quality source of nutrition for tadpoles. So it could be providing additional resources that could help them fight infection. But what I was really interested in is what does this do to their gut microbiota? And so given this background in the system, we set out to understand what effect these different environmental conditions had on the gut microbiota, and then in turn, how this might impact host pathogen interactions in this system. And the reason we were so interested in the gut microbiota is because in other organisms, it's been shown that the gut microbiota influence host health through a variety of mechanisms, which I've listed here. And particularly relevant for tadpoles, we know that bacterial fermentation in the gut results in the production of short-chain fatty acids. And these are a highly nutritious resource for tadpoles. But the other cool thing about them is that they're capable of crossing the intestinal epithelium and traveling through the circulatory and lymphatic systems to bind to receptors in the nervous system and the endocrine system, and therefore are capable of influencing host metabolism, host physiology, and all kinds of things. However, the amounts and types of short-chain fatty acids that are being produced is going to be highly dependent upon what bacteria are present in the gut, as well as what the host is eating, right? So we set up an mesocosm experiment with the idea of sort of indirectly manipulating the gut microbiota through changes in diet. And then we wanted to look at what effects this would have on the host under high and low salt conditions. So we used approximately 50-gallon mesocosms, which I think is about 200 liters. We set these up with tadpoles. We added dried leaves for cover and substrate, and we fed all the tadpoles a basic diet of boiled alfalfa. For the diet treatment, we supplemented half of the tanks with chlorella algae, which is a fairly easy to obtain and also known to be highly nutritious resource for tadpoles. And then we added road salt to half of our treatments to increase the salinity, to elevate the salinity. We let the tadpoles develop until they were approaching metamorphosis. And then we took individuals from each of our treatments and brought them into the lab in little individual containers, and then exposed half of them to ranavirus during that period of high vulnerability, right? We took a lot of information from these tadpoles. We weighed and assessed their developmental stage before we put them into the exposure treatments. And then we did this again 6 days after infection, which is when you expect ranavirus levels to have increased, but not to the point where it's going to kill the tadpoles. We dissected out the livers to assess viral load. We dissected the kidneys to get corticosterone concentration, and we took their guts to characterize their bacterial communities. All right, so first I'm going to share what happened to the hosts. So we found that increasing salinity and supplementing the diet with algae enhanced larval growth. So this is kind of what we expected, right? So in the salinity treatments, we could expect there may have been some natural increases in algal abundance in those treatments, but the algal supplementation also appeared to have a strong effect on growth and an even stronger effect than salinity. But there was a drawback in that these animals developed more severe ranavirus infections. So instead of having the algae make them less susceptible, it seemed to be making them more susceptible to infection. So this was a little surprising. But then the question is, how did they respond to that infection? Or the question then becomes, how did they respond to that infection? And so first, when we looked at larvae from the basic diet, we saw that they responded exactly how we expected. So here on the top, I'm showing you the change in Gosner stage over that 6-day period of exposure. And below, I'm showing you the change in weight. And in orange here, we have animals that were exposed to ranavirus, and then in black are our controls. And so what you can see is that the animals that were exposed to ranavirus increased their development, their developmental stage changed more, and they lost weight over that period, especially in this high salt treatment, right? And so this indicates their HPI axis was activated, and they were inducing a stress response. But we didn't see this for those animals that were fed algae. So in this case, animals that were exposed to ranavirus didn't accelerate their development or lose weight. They just kept plodding right along. And so even though they had high infections, right? And so this was a really interesting result. And what was nice was that when we looked at their CORT levels, we saw that they also had much lower levels of CORT. Animals whose diets were supplemented with algae had much lower levels of CORT. So again, supporting that algal supplementation is somehow suppressing activation of that HPI axis. And so how did these outcomes correspond to variation in their gut microbiota? First, we can take a look at salinity, and what we see is that increasing salinity resulted in higher relative abundances of proteobacteria, and this may indicate that there's some dysbiosis happening. So higher ratios of proteobacteria are often associated with imbalances in the gut. I'm going to kind of speed up a little bit here. Whereas algae, on the other hand, had a very different effect. What it did was it decreased the relative abundance of bacteriodota. So you're same as me, you're like, what does this mean? Which is most of what I do in my research. But because we really know very little about these organisms and what they're doing. But it was interesting. And then the other really cool thing is that we saw the same effect even in the high salinity treatment. So animals, regardless of whether they were exposed to high salinity or not, had very similar gut microbial communities that had very similar structures, right? And so again, this suggests, one, that algae has sort of a rescue effect on the gut microbiota, and it overcomes the effects of salt, which was cool. And it also nicely explains why all of our tadpoles, regardless of salt treatment, whose diets were supplemented with algae, had those lower CORT levels and that reduced response to rhinovirus infection. Okay. When we dug a little bit deeper into this, maybe this tells us a little bit why. So Bacteroidota and Firmicutes are two really important components of the gut microbiota of lots of different organisms. And they're also associated with the production of very different kinds of short-chain fatty acids. So in the animals that had a basic diet, we see more Bacteroidota, which are associated with the production of acetate and propionate, whereas in those animals that were fed an algae diet, we see higher abundances of a Staphylococcus in the Firmicutes, which are more associated with the production of butyrates. And Staphylococcus in particular are interesting because they have been shown to enhance the extraction of nutrients from they have been shown to be very effective at extracting energy from foods and other organisms. And so what we think could be going on is that these algae-induced shifts in the gut microbiota could be leading to very different kinds of resources available to the host to fight infection, but then also very different kinds of short-chain fatty acids that could be interacting with the neuroendocrine system to regulate their response to infection. Okay, so to summarize here, does variation matter? First, I just want to acknowledge that response to disease is determined by many factors. So I've showed you a few of those factors here: developmental stage, salinity, diet, maybe also microbes. And in particular, we see that even subtle differences in composition and structure may have consequences for the host. But we're a long ways off from understanding the exact mechanisms of how these things work. I think the work that I've showed you today kind of emphasizes some of the next steps we can take to understand this a bit better, but we still have a lot of work to do. And then finally, just to kind of bring this back out, beyond frogs, even though frogs are awesome, as we are all aware, wildlife is experiencing unprecedented environmental change at the hands of human activity. So whether that's exposure to novel pathogens or increases in environmental pollutants or changes in climate, right? However, several recent reviews have highlighted the potential of microbial symbionts to, in some ways, shield their hosts from some of the worst effects of these changes through the ecosystem services that they provide. And this could allow them to perhaps persist or maybe even adapt to some of these novel environmental conditions, which to me is really cool to think about. And so hopefully the research that I have presented to you today gives you some insight into just how responsive these microbial symbiont communities are to environmental conditions, and also during which life stages and under which conditions bacteria can best be incorporated into these symbiont communities. And then finally, what this may mean for the host itself. All right, I'm done. It's a little long, but I'm done.
Thank you very much, Myra. So it starts the time for questions. Anyone? A couple.
Thank you, very interesting. I'm particularly interested in the dispersal work you're doing. So I guess you would expect that if you had like a 3 species where you compare the skin microbiome, if they came from the same pond, then they probably got more similar communities than if you compare populations from different ponds, right? Have you looked at that at all? Yeah,
we have. So you get the same distinct clustering. So even within a pond, if you sample different species, they still cluster out separately. So there's something, probably something about the host environment that is still having a relatively strong effect.
But there is a pond effect as well. So that there is kind of the fingerprint of individual ponds, or not?
I don't know if I can say that off the top of my head. Yeah, if I don't think I have done the work to completely tease that apart, the strength of the pond effect versus the strength of the species effect. My guess is the species effect is stronger.
Right. And what about dispersal mechanisms for those bacteria? I mean, to what extent do you think they're waterborne dispersed, and what extent there might be aerial dispersal, and the chances of dispersal during the terrestrial phase compared to the aquatic phase?
You mean how stable are the communities for kind of the terrestrial species versus aquatic species?
I mean, how those bacteria actually colonize, like in later life, let's say, if an adult that's in a terrestrial phase of its annual cycle can be colonized by new bacteria that's not present. So to what extent, would there be aerial dispersal of these bacteria compared to movement of their propagules within the aquatic environment?
I think that the aquatic environment is really great for homogenizing these communities, probably, and you could probably get assembly dynamics going in much more different directions in sort of more terrestrial systems. But we also know that these communities are really dynamic and responsive to short-term variation in environmental conditions. So we have, for example, the red-eyed tree frogs And the pantless tree frogs we've sampled seasonally, and we've sampled, after rainfall, and we do see, sometimes dramatic changes in their microbial communities. So I think there's a lot of turnover in these communities in general. Yeah, and so actually we're looking at some data now where we found consistency over time, which was really surprising for us. And that was in the larval community. So these are samples of tadpoles that we've taken over a 3-year period. And, no. These are, we've sampled the same ponds over 3 years. And their tadpole communities, the communities of the tadpoles are similar across time. They're different between ponds, but they stay consistent across time within a given pond, which we found really interesting.
Cool.
Yeah.
Okay, we have a question in YouTube. Mylène Mariette is asking, is saying, thanks a lot for an amazing talk. Can the host cord levels influence which bacteria can persist in an individual and thus potentially worsen disease impact?
Sure. So I'm definitely not a physiologist. Erica is my collaborator on this, is much more of the physiology expert on this side of things. But one thing that we thought about that's really interesting is, these animals are developing these more severe infections, and it seems like they're doing well, but we don't know what they're carrying with them after they metamorphose and what the sort of long-term impacts. If we had carried that experiment out through metamorphosis, would they have survived? We don't know. And also, could they be transmitting the virus more, retaining it into the next life stage and then become essentially reservoirs of the virus that could transmit it to the next generation. So there's definitely lots of interesting, I don't think that it's necessarily all good for the tadpoles.
Okay. And we have another one. Yes. Elena Angulo is saying, thank you for the nice talk. Were the algae sterilized before feeding the frogs? If not, could the algae microbiome work as a probiotic booster in the beneficial bacterial community?
Ooh, that's an interesting idea.
It's a triple interaction.
I know, exactly. I mean, yeah. So we did not sterilize the algae, and these were done in outdoor mesocosms. So there was all kinds of stuff coming in here that could potentially be, these were very natural systems. And so I think that's definitely another layer to consider in kind of future work in this system. Yeah, super cool.
Totally. Anyone from the room?
Hi, thank you. It was a really good, wonderful talk. So I was wondering, for both the case of the chytridiomycosis and the ranavirus, if you have studied what is the microbial community before and after?
Yeah.
So how does it change? Because, for example, it's interesting, Pseudomonas also changes a lot in malaria infection in humans. So it's involved. Yeah, so humans that are infected or not with Pseudomonas have are infected, are more likely or not to be infected with malaria. So I was wondering if you know how it changes.
Yeah, so bottom line is no, it did not change at all. So we definitely saw big effects of bringing the animals into captivity that may have masked some of the effects of infection that we might have observed. The other thing to know is that all of the species, actually all of these species here are generally relatively resistant to chytrid, and they seem to persist with low infections in the wild. So one of our one of our thoughts about why we might not have seen a response to infection is that these communities are already pre-selected to resist as much as possible. So yeah,
So even though they are like, well, because they are pre-selected, they still get infected.
They can still get, well, that also varies. So this species right here, we never saw high infections in them.
Nice.
And so I think there's probably some intrinsic immunity or something about the frog itself that helps it resist chytrid. But these other species, oh, and this species too was the same way. These other species, yeah.
Yeah, I mean, it interacts with MHC and all that. So.
Yeah, and then work that folks have done in other labs with other more susceptible species or different species, we do see changes in the microbial communities in response to infection. So it just didn't happen in the species that we chose, but it does happen, yeah.
Thanks, Myra, by your interesting talk. Well, my question is related to, have you some information on microbiome community and divergence at the metamorphic climax? As you know, metamorphic climax implied deep changes in good entirely chain, the good, and also the tegument. So also is the moment where more infection by BD, for instance, a lot of casualties and mortality increase during climax.
Yeah.
So what's the information at this period?
Yeah, so we actually have a data set where we're looking at that now. And so it's the same data set that I was mentioned to you, where we've sampled larvae from a variety of ponds on Vassar's campus over time at different developmental stages, different ponds, different years. And we do see a succession with developmental stage. And so I don't know how familiar you are with Gosner stages, but we've got up to stage maybe 44, so that's like when their arms are popping out. And we do, it seems that we, in that, those late developmental stages, we get much more variation in the microbiome, and the microbiomes become much more dominated by like a single phyla, or but it's different across different individuals. So I think there is sort of a breakdown of the gut bacterial community, at least as they near metamorphosis, or the kind of integrity of the gut community as they near metamorphosis. And yeah, I think that could be doing some interesting things there in terms of susceptibility.
Thank you.
Okay, sorry, but we have the time. I will ask you a question afterwards.
Okay.
Thanks a lot, Myra, for this very interesting talk. And thanks all for coming. See you next week for the last EBD seminar of the academic year. See ya.